So now we're moving on to the nerve impulse. How do these neurons actually send a signal? How do they talk and listen to each other? Let's dive into the nerve impulse and kind of piece it apart to understand what's going on physiologically when we're sending a signal down a neuron. So the first part of the nerve impulse is called the resting potential. The resting potential is when the axon is not sending a signal. We're not talking to another neuron during the resting potential. When we're found during resting potential, when the axon is not sending any sort of nerve impulse down the line, there are more positive ions outside of the membrane and fewer positive ions inside of the membrane. Because there's fewer positive ions inside, the inside of the axon has a negative charge. There's two particular ions of interest we're looking at as well. There's sodium and potassium. During resting potential, sodium is mainly outside and potassium is mainly inside. So we have a negative charge, negative 70 in this case. Sodium is locked out and potassium is locked in. And so that's what we see here, right? So here's our axon, right? The membrane is this yellow tube. We see that there's a negative charge all along the entire axon on the inside and a positive charge on the outside. If we look at the inside of the axon, it's mainly potassium and the outside is mainly sodium. Keep in mind, when we look like this during the resting potential, we're not sending a signal down the axon. Now comes the action potential. This is where we're actively trying to talk to another neuron. This is where we're sending a signal, we're sending a nerve impulse down the line. And in order for this action potential to happen, we're going to have some rapid changes occur. They're going to allow for this message to travel along the axon. So we can split the action potential into two parts: depolarization and repolarization. During depolarization, there are gates for sodium that are going to open up, which means all that sodium we found on the outside is going to flood inside of our neuron. When we flood in the sodium, look at the charge. It has a positive charge. So if we flood in something that has a positive charge, we're going to change the charge on the inside of the axon. Remember, it was negative 70. It's going to change to negative 55 up to positive 35. So we've gone from a negative charge inside to a positive charge inside. And that's really one of the big things that's going to allow us to communicate, allow this message to travel along the axon. So again, depolarization is the first half of the action potential. We open up the sodium gates. We allow sodium to flood in. Sodium has a positive charge, and so we're going to change from a negative to a positive charge on the inside of our axon. Right, and so here we're seeing that change in the process. We're seeing some sort of stimulus, something that's telling us to open these gates. We reach that negative 55, that's going to be our threshold. That's going to kind of push us to continue. If we don't reach that threshold, then the signal pretty much stops. But if we do reach this threshold, we can keep on going. And we get to that positive 35. Now repolarization is the second half. Repolarization is when we open the potassium gates and we let potassium out. Potassium also has a positive charge, so if we let the positive potassium out, our charge is going to go back to a negative. And so we're seeing this wave of depolarization, where we become positive on the inside, to repolarization, where we become negative on the inside, happening in a wave-like motion all the way down the axon. So it's not an entire axon going through this process, it's sections of the axon are going to go through this process. Now, if you paid attention, or if you remember from resting potential, remember that mainly sodium was on the outside and mainly potassium was in the inside. But if you've been keeping track of the ions at this point, you've noticed that we've swapped them. By the end of the action potential, sodium's on the inside and potassium's on the outside. How do we get back to resting potential? In order to get back to resting potential, we're going to have to pump potassium back in and the sodium out. So we're going to have to swap the ions back to the starting point to get back to our resting potential. That's called a refractory period, where we have to kind of reset ourselves before we can send another signal down the line. The synapse is where we communicate from one neuron to the next. The synapse is a gap. So these neurons that are talking to each other don't actually touch each other. There's a small space between them. So the gap that we have between the two we call the synapse. The presynaptic membrane belongs to the neuron that's sending the signal, and the postsynaptic membrane on the other side of the synapse belongs to the receiving neuron. In order to communicate the signal across this gap, instead of using a change of ions like we did throughout the axon with the nerve impulse, we're going to use a chemical instead called a neurotransmitter. For example, acetylcholine, dopamine, serotonin. We've seen acetylcholine before. We saw that when we talked about muscle contraction. We won't get into specific neurotransmitters and what they're used for. We're just going to have the general idea of what a neurotransmitter is. It's a chemical that we can use to send a message across the synaptic gap or the synaptic cleft. Now, these neurotransmitters are going to be stored in the axon terminals of the sending neuron. So you remember that long axon, all the way down at the very end, we had those little like finger-like projections. Those were the axon terminals. And so we have vesicles hanging out on the inside of the axon that are waiting to fuse to dump those neurotransmitters into the gap so they can travel across the space. So the nerve impulse reaches the axon terminal. Calcium ions are going to come in, and they're going to tell the vesicles to fuse with the membrane to dump the neurotransmitters into the synaptic cleft or the synaptic gap. And the neurotransmitters are released into that space where they can then bind with the postsynaptic membrane, the receiving neuron, to tell that neuron either, hey, go ahead and send an excitatory signal, or they can inhibit that process. So what we see if we look really close, again, so here is our axon terminal. Right? It looks like it's touching, but if we zoom in, we see it's not actually touching. There's a space between them. Calcium comes in, is going to tell these vesicles to merge with the outer membrane, and when they do, they spill their contents into this open little gap, right into the synaptic cleft or the synapse. Those neurotransmitters can traverse across that gap and then bind with a receptor, and then this receptor will tell the receiving neuron to do something or to not do something in response. Integration is what happens when you have many signals being accepted by one neuron. So we've been talking about it in a very singular format. One neuron is talking to another. But in reality, a single neuron is going to get many signals from many other neurons. Multiple neurons are connected together. And so how does a neuron decide what to do when it has all of these signals coming in and some of those signals probably conflict? Integration is the answer. The summation of the inhibitory and excitatory signals are going to tell our receiving neuron what to do. If there's more inhibitory signals, then we follow those instructions. If there's more excitatory signals, then we'll follow those instead. So here we see this cell body, we see this neuron that has all of these different axons reaching it. So multiple neurons are communicating with this one we see front and center. And we see Here's our resting potential. Here's our threshold. We have to pass that threshold in order to send in that excitatory signal. We have a lot of inhibitory signals. We have some excitatory signals. And if we integrate, we take the summation of both, we don't quite reach that threshold. And so we won't send an excitatory signal here. So that's all we're doing. We're looking at what is the summation, what's the sum of all the signals we're being sent, and who has majority, right? Who can we reach the threshold or not? If we don't reach the threshold, then inhibitory wins.